Good morning.
Good morning.
Morning.
How are you today?
Ah, nice.
Where's the rest?
Yeah, late.
Those of you who just came in, can you see the link to the paper in the chat?
Otherwise, I just share it again.
Yeah, I didn't get it yet.
Okay.
There we
Thank
go.
you.
I'm just going to share it over on YouTube. Today's paper. And
Should I already share my screen maybe and see if that works?
Just going to make some people co-hosts in case things go wrong. There we go, Evie, you're on the list this week.
Yes.
Great. There we go. Diffuse the responsibility.
Yeah.
Fantastic. Well, welcome to Neuroccino 931. As you know, we try to discuss exciting science every week, and everyone is welcome to come and present a paper they came across and thought was exciting. Come and tell us why you thought it was exciting and what you liked about it. And then we discuss it to the best of our knowledge. Today we have quite an exciting paper that I actually came across and thought it was super cool. And then someone actually volunteered to present it. It's going to take us out of our comfort zone a little bit this morning, because it's about genetics, machine learning, genetics, all the fun things. But I'm quite excited about the discussion that we're going to have afterwards.
Yes, I try to keep it light and entertaining.
That's the spirit. You already heard her and saw her. That's Barbara. Thanks for sharing your screen. She is a postdoc here at the Max Planck Institute for Psycholinguistics in Nijmegen. And she's working in the group for genetics and language with Simon Fisher. So today is going to be all about genetics. Great to have you. Thanks for volunteering. If you could show your paper in full screen, please.
Should be full screen, at least that's what I think.
If you can zoom in so we
exactly.
See the full paper, it's easier. Oh, perfect.
There we go. Now I think it's massive. And
lovely. Over to you.
Yes, so hello everyone. I'm quite excited. That paper came across my Twitter feed, and I think we already talked about it in the lab a tiny bit. But as always, it ended up on a pile and I haven't read it. So when Stephanie invited me, I thought that's a great chance to read that paper actually, because, well, it's about doppelgangers. So I thought that's super exciting, but also it's kind of like a bit of a different take, or quite a, I think, elegant way to discuss the nature versus nurture issue. And beforehand, quite often what was done in genetics, you did this with twin studies. So you had twins that shared the genetic background, but then potentially they're raised in different environments. And so that's how we kind of try to answer this question of nature versus nurture. This paper now does a complete different way. They look for completely unrelated individuals that look alike, and then have a look if they actually share genetic components. And I thought that was super intriguing, and that's why I wanted to talk about with you today about this. But it's genetic-y. There is not that much neuroimaging, but I think at the end, I want to give a bit of a try to push this discussion towards neuroimaging, and if we could use this, but also have a bit of a ethical small point, because I think that's quite important to think about this, and they raised this, or at least talked about this at the paper. So first of all, what they did. So I just, whoops, let me go to the paper. Okay, this is now because it's on massive, it's more than one thing, but here we have the thing that I wanted to show you. So here you see the figure 1, and what they did, or how they got their doppelgangers. It was an artist that basically started to collect images worldwide of people that are doppelgangers, And they chose 32 Canadian lookalike pairs and then used like facial recognition software to kind of like objectively actually match them together and be sure that it's not just that we think they look alike, but that you have a measurement. Here, I think in 1B exactly, you can see one of the algorithms and all the facial morphology points that this algorithm uses to identify or like to match people together. And then in 1C, you can see the 3 different nets that kind of like were used. And of these 32 pairs, 16 were identified in all of the 3 algorithms that were looking quite similar. And in D, you have the kind of objective similarity scores that rank between 0 and 1. And you see like for each algorithm, kind of like in light blue, that's monozygotic twins. So like that is a twin sample, so they should look alike and they score quite high. But then in this like pinkish color are the lookalikes from the database. And they actually are either on the same level as a monozygotic twin or for like the MatCon net actually a bit higher. And in E, you see an actual example of how these participants or like this lookalike doppelgangers look like. And I found that fantastic and amazing how similar they actually are. And then once they had like their pairs of lookalikes or like the 16, they did 3 kind of like steps of analyses. You can see this in 2A. So first of all, they did basically like genotypic arrays. So they looked at SNPs that are common variants. So for everyone that doesn't know what a SNP is, so it's literally a single nucleotide polymorphism. So like one base that is different, but we all have them. They are common. So it's nothing that is rare. But they also looked at epigenetic changes, so DNA methylation. So that is not a change of like the DNA sequence itself, but still influences gene expression. And then they looked at the microbiome of these participants and just had a look if similar looking people actually also share similar genetic backgrounds. And here in 2B, that looks quite confusing at the beginning, but basically it's just like a heat map where they try to cluster the genetic information with Bootstrap. So they used here the information of the genome-wide SNP genotyping arrays. And of these 16 lookalike pairs, 9 clustered together. So they were deemed as ultra lookalikes. But obviously, like this genetic analysis could be biased by 2 things. It could be that these participants, without knowing, are related to each other. So that sometimes happens, but if you have the genotype information, you actually can calculate a score that tells you how related some participants are. And that's what you see in 2C. So that's the king cryptic relatedness. You again have monozygotic twins, and it's known that monozygotic twins have a kinship score of 0.5. And then you have a look at the LAL is the lookalikes and then the non-lookalikes, and they have a quite low kinship score. So indeed, they're not related. I think in the paper, they found just 1 pair that was potentially related up to the 3rd degree of relatedness. But otherwise, they share genetic similarity, but not in a case of being actually related to each other. And what they then also did, they had a look at ancestry, because obviously if you come from a specific ancestry, that might also explain why you overlap quite nicely. But in the end, also ancestry could not explain why they are sharing this genetic similarity. And what they then had a look at the genotypic level is they looked at the number of shared SNP positions. So if one pair was, for example, a certain position heterozygous for a certain SNP, the other individual in the pair had to be heterozygous as well. And in the end, they found that they had a higher number of shared SNPs compared to if you look at a random sample. Even if you take into account the ethnicity of this participant. So these people in the end definitely shared some genetic similarity. And when they in the end looked at the SNPs and how they're related to certain genes, you can do something called a gene enrichment analysis. So basically you look in which pathways or similar these genes are enriched, and what they found here. So that's done in the biological processes, so in D, in molecular function, and in a cellular compartment. You actually see that you found an enrichment in anatomical development, in cell junctions, so tissue morphology. So I think it's quite clear in which biological processes these genes were related to. But I think the biggest point that they made is at some point that they looked also at genome-wide association studies for facial morphology. So you get all the SNPs that are in some way related to making up facial features. And there they found really the highest hit, basically. So of this nearly 20,000 SNPs that were similar in these lookalike pairs, the majority were found in like pathways or in genes that somehow define how a person looks like. I think on one side, that is a bit of a given because we have pairs that look alike. So we find also kind of like an enrichment in genes that make up the face. But on the other side, I think if you think about this, that actually is quite a big thing, that people that have nothing to do with each other, that simply just share same facial features, actually have the same underlying genetic components that make up facial features. And I think that can be quite broadly applicable for a lot of different things. But they didn't just stop there. So it's not just that they looked at the genetic or genomic component. As I said, they also looked at epigenetics and microbiome and had also looked if anything was shared there. Here they found actually not that much information. So that's why I don't show the figure. For like epigenetics, so DNA methylation can also be used to determine the biological age compared to the actual chronological age. There they mentioned that it is possible that the biological age is a bit similar in these lookalike pairs, but actually like they didn't find that kind of like massive points. And then I think another very important fact is that if we go away from complete genetics, they also had like a massive study and looked at different biometric and lifestyle analysis. So here in 2A, you can see what actually they looked at. They looked at exercises, if people have pets, what handedness they have or family status, and then used something called Euclidean distance between the individuals, and also found that the lookalike pairs shared actually lifestyle similarities compared to the non-lookalike pairs. And I think that is quite big, that not only is your genetics, like the facial morphology, similar, but for example, they're also a bit more similar if you look at smoking. And I think there starts to be that everyone can see, I think, the bit of applicability of what you could use this for. Obviously, this is a very tiny sample. So we're talking about like 16 or like 32 in total pairs at the beginning. So I think that's what they also mentioned massively in the limitations of the study. But what they say, like that's also here in red, like these findings provide a molecular basis for future applications in various fields, such as biomedicine, evolution, and forensics. So I mean, that is like the beginning. And I think I would absolutely want to see this replicated in a bigger sample to be sure that this is really the case. What I'm, for example, missing, and I mean, that's probably because of the sample was quite small and that was like an independent study, is what happens to neuroanatomical features. And I guess like that is a neuroanatomy journal club. So I think everyone would be quite interested in. We had a bit of a discussion if that actually is possible. I mean, you have 23andMe, you have UK Biobank. There might be lookalike pairs in it, but I think here we come to a bit of the ethical dilemma that I don't think it's possible to have also an imaging, like a picture database, because we even deface our imaging data sets. So I don't think that is possible at all. But then I thought about, and I think that's one of the first discussion points where I would like to just get your thoughts on that. If we could do the same, instead of facial features, use neuroimaging features. So have similarity in any kind of neuroimaging traits and then see if that also relates then to genetic similarity. I'm not sure if it's possible to simply just use T1 weighted scans or diffusion data and put it through an algorithm and see how similar these participants are, and then use that data to look at scanning. And then I think another point is then the ethical implication, because I think it was like written down here. let me see if I can. Our results are, but I just have to move you guys around a bit. Our results are germane to the ongoing efforts to predict biometric traits from genomic data and the diagnosis of genetic disorders using facial analysis technologies. So I don't know, when I read that sentence, I just get a bit of red flags in my mind, because I think there was recently a post where I think the police used DNA phenotyping to solve cases. And I think it's a bit of a slippery slope sometimes. So I think you have to be really big aware of what you can predict and what you can't and how that's used. But also I think if you diagnose genetic disorders just by looking at a face, that also I think could be misused in a certain way. So I think it has to be always quite clear what this can do with you. But I think like here's a bit of the difference. Like if we use neuroimaging traits and then look at genetic similarity, that is something that would be applicable in clinical. But I think if it's going from facial features that can be just collected everywhere you walk around, I think that might be a bit of an issue. So I was just wondering what you think of this idea. So like this nature versus nurture different way of doing it. So instead of looking for similar people or like related people and then checking kind of like the different lifestyles, but now turning the way around and using completely unrelated people, but using similar phenotypic features to look at their genetics.
Awesome. Thank you so much. The paper is just as exciting as I was hoping it would be. And I love that you already had many pointers in terms of discussion points. So first, let me see if there's any questions in the room. And whoa. Okay, hands are up. Hands are up. Whoa. I have no idea who was first. I can see Hervé first on my screen. So there you go.
Yes. So my question is maybe more a comment based on what you say at the end of your presentation on the facial recognition, because it reminds me a discussion we had for the Imagen consortium, where we had a project for looking at neuroimaging data, so using the T1 MRI data, so using the T1 to reconstruct the face of the subjects to get some of the idea of looking at this phenotype and to look at if some disorders can be predicted based on this. And so we had a lot of discussion because that raised a lot of issues on recognition and identifying the subjects. And at the end, I don't remember what happened, but that one of the, I think the guy in the room was saying that, but the face, if we can use the face as a real phenotype for disorder, it will be easier as compared to neuroimaging data to collect data. So the point was, yeah, if it is a good proxy, maybe we can use it. After, yeah, we need to be careful about the ethical issues that can raise with the use of this kind of phenotypes. That is a comment. I had questions, but I think you replied a lot to a lot of them. Maybe to get a little bit deeper on one of them, I was wondering if they look at, but I think the figure 4 was answering this question. So the relation between the facial similarity and other phenotypes like size, weight. I think that
yeah,
I think
exactly.
the figure 4 is looking at that.
Yeah, they definitely looked at that. I mean, that is just like, I think, a similarity, like Euclidean distance between the individuals from a pair versus a non-pair. That's, I think, 68 different metrics was what they looked at. And I definitely like weight, height, smoking. So also behavioral things were included in this. And it looks like that people that look alike also share some, I think, behavioral traits with each other, which would be big. I mean, that is like massive. But it might just be that, for example, the SNPs that are important for facial features also have any kind of implementation, like links to behavioral traits, or it's simply that just an environmental thing that people that look alike do the same things. I don't know. I think that is like what actually is causing each other, I think it's a bit hard to say.
Yeah. Okay, thank you.
Thank you. Chris.
Yeah, thank you for presenting this paper. It's really interesting. One question, comment, I guess. So it's, when I heard the smoking part, that they had similar behaviors towards smoking, that makes me think that it could be, instead of using it as what you were saying, like it can be scary to use it to diagnose just on the face, but maybe use it like you see a diagnosis, and then you see what population according to what features have these different behaviors, not only about smoking, but because smoking, it makes me think of addiction, and addiction is a massive field of research, and it's really important for a lot of people to try to find solutions. And also, would there be other features, because the facial features are the ones we see, but you were mentioning the MRI, if we could find some similarities in the brain and be like, oh, we can relate it to similarities in the genome, and use the same kind of grouping methods to say, oh, we can relate this feature in the brain to this disorder or this disease. So and then we can relate it to genes. So we could have a whole thing to screen people. Like we would see a feature that is maybe easier to spot than other things, and also it might allow to reduce the samples that we need, because now the DNA analysis, what they need is a massive amount of data, and now just trying to find like similarities. Oh, these kind of genes, they are increasing the risk of this disease. But if we had other domains that were linking to small sequences of genes, maybe that would help reducing just improving the accuracy of the predictions.
I mean, definitely, like the thing is, what I'm still wanting to see is if that replicates if a bigger sample is used, because I mean, now you see this in 16 people or like 32. So in the end, they say themselves, they need to like just like generalize this and then see if the effect is like constantly there. And I think with imaging traits as well, you probably would need a lot of samples to kind of like create something similar, like this facial recognition software that really clusters them. So I don't know how many imaging features you would need to be really sure like this is more similar than another brain that is randomly picked. But still, like I think it's intriguing. But on the other side, what you also have to take into account, like the overlapping SNPs, it were I think 20,000 SNPs were identified in this lookalike. So that's like quite a lot, and they might be involved in a lot of different traits. And I think you just need to dig a bit deeper yet to really use this as a diagnostic marker. But I think it's just like the idea of turning the wheel around and instead going for twin studies that are rare and then finding twins that are raised in different environments. I think that the other way around, to have unrelated people but correlating on a phenotypic level and then going on to the genotypic level, that would be intriguing.
Yeah.
Just, I think it's like a nice idea to start thinking the other way around.
Yeah.
But yeah, I think maybe like you don't need as big sample sizes, but again, in the end, if you then make inferences about any kind of genetic disorders, then again, you probably might need bigger samples again to be quite certain.
Yeah. Thank you.
Victor.
All right, thank you for the great talk. I was just wondering, because they have the algorithm, the automatic face analysis algorithm, they know about each feature, each face feature, right? And did they only do the paired experiment between the lookalike, or they also say, okay, all the people who have the same nose, all the people who have the same eyes, all the people, blah, 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 and try to replicate and find if there were constants in these kind of things?
No, I think they just literally looked at the lookalikes here. So literally they just used the algorithm. They had their like 32 selected pairs, and then just looked at the overlap, like the lookalike pairs that were identified in each of them. But I think that might be true. That plus that there, I'm not sure. I think the Microsoft algorithm where you saw the facial morphology features, there you definitely probably have information in which features they like are similar. But I'm not sure if all the other two work in the same way. But that might be then nice to see like what it's like related to. I think the nose and like the frontal facial features were like the ones that had the biggest impact. But otherwise, then I think I think also for like dissecting that, you again need a bigger sample, because now you have like generally overall 16 lookalikes, but then if you go for like a certain like eye trait or certain this, then in the end you just might end up with one that maybe clusters specifically in this category.
True. But there was a follow-up comment to this question, because I was thinking, if we start to do that and somehow manage to have some kind of predictive value of what you will look like, that really sounds like the beginning of eugenics and stuff like that, where if you do, if you give this technology to countries like, for example, I just think of China, where there was, at least before, not that much, not anymore, but there was a lot of pressure on parents to have only one child. And so they wanted the perfect child and everything. And so this kind of thing, when they had, if you could have given them the choice of the feature of the child, if they could, this kind of thing could really have big slippery slope into dangerous selection and eugenics and stuff. But
well, yeah, that's why I think I wanted to raise this point, because I think there you have to be super careful in what this would mean and could do. Because like I don't know how long it was ago, a month or so, like the tweet pop up that like the police used DNA phenotyping to predict how like assault person like looked like, or like someone that assaulted someone, and they used, I think, even pigmentation or something, and there was a bit of an outcry on Twitter that that's not exactly how it works. So I think you always have to be quite aware what you can recognize, what you can't recognize, and what you can predict and what you can't. So I think it's just like always with a bit of a grain of salt to make sure how much power it has and whatnot.
True.
Should generally, I guess, have more emphasis on neuroethics, genetics and ethics. That should be a column in the curriculum.
Yes.
Because sometimes I feel like we're getting ahead of ourselves, and we use the amazing methods and tools that we have available at the moment, and we do them because we can and because it's exciting and we find out new knowledge, but then it's worth taking a step back and looking at the wider implications of that kind of research.
But like from my side, also like a question to the more neuroimaging community. Do you think like the possibility to use neuroimaging scans is possible to get like a global metric of similarity and then use this as a trait instead of facial features? I was just wondering if that's like...
I mean, I'm sure it's possible technically, but the brain is a 3D thing, so it's really hard to get easily this kind of thing, because with the lookalike, we use our innate capabilities to check if we can actually to recognize different people, and so this is something that we are really very well trained to do. For brain, we'll have to create an algorithm that does it for us, and that would not be easy. I think possible nowadays with current technologies, with all these AI and that are able to do a lot of things like this. It's possible, probably, but I think I don't know how hard it would be.
Jaco also wants to say something.
Yeah, I want to like chip in this conversation, although Victor left, so maybe last moment. I just wanted to add, because you said like that for recognizing faces, we do it like kind of naturally, right? But then in this paper, they had an initial sample of 32, and then they downsampled the sample to 16 after using algorithm to match pairs that really looked alike. So for brain imaging or for like other type of phenotypes, like we shouldn't start by like looking at the things that look more alike, but we could already place them in a space and then compute some sort of like similarity or dissimilarity metric, Euclidean distance or whatever, and then not need to downsample it again, having a kind of objective metrics from the beginning. So I don't think it's like so crazy as an idea.
It's actually something that we're doing in the lab.
Yeah. That's why I thought I might get some.
Just putting it out there.
That's why you thought.
Yes, I totally agree that you can put it in a space and compare. It actually works. We've seen the preliminary results, which is quite amazing. But I guess that's a wider discussion that we should have maybe over a coffee tomorrow, and see where we can take it and how we can combine it with imaging with genetics and genetics with imaging for the different projects. That's all the time we have. Thank you so much for coming this morning, for presenting a very exciting study and taking us a little bit out of our comfort zone. We shall see you next week again. I don't think we have a volunteer for next week yet, so if you come across some exciting science and want to present it, do let me know. Other than that, I wish you an exciting week in science and see you next Monday. Bye.
Bye.
Bye.